Chapter 4: Antibody Structure and the Generation of B-Cell Diversity. In the first panel, we see a resting B cell. These cells have membrane-bound immunoglobulins, or antibodies, on their surface, which function as receptors for antigens. When a bacterium or any foreign antigen is encountered, as shown in the second panel, it binds to these surface immunoglobulins. This interaction activates the B cell. The third panel illustrates what happens next. The stimulated B cell undergoes a process called clonal expansion and gives rise to plasma cells. These plasma cells are specialized to produce and secrete large quantities of antibodies that are specific to the original antigen. These secreted antibodies play a crucial role in neutralizing and clearing the pathogen from the body. This is a foundational concept in understanding how our immune system defends against infection. Antibodies are composed of polypeptides with variable and constant regions. Antibodies, also known as immunoglobulins, are Y-shaped molecules made up of 4 polypeptide chains. 2 identical heavy chains shown in green and 2 identical light chains shown in yellow. These chains are held together by disulfide bonds to maintain the antibody structure. The tips of the Y, labeled as the N-termini, are where antigen binding occurs. The base of the Y, or C-termini, helps anchor and stabilize the molecule. In the lower panel, the antibody is shown color-coded to highlight its functional regions. The variable regions, in red, are found at the tips of both the heavy and light chains. These are the parts of the antibody that recognize and bind to specific antigens. The constant regions, in blue, form the backbone of the molecule and determine the antibody's class and function within the immune system. The hinge region, located between the arms and the base, allows for flexibility so the antibody can adjust to bind multiple antigens more effectively. This slide demonstrates how an antibody can be enzymatically cleaved into distinct functional fragments. At the top, we see an intact Ig antibody. When treated with a protease enzyme, cleavage occurs at the hinge region, the flexible segment that connects the arms of the antibody to its base. This cleavage results in the production of 3 distinct fragments, as shown in the lower panel. 2 Fab fragments, short for fragment antigen binding, are released. Each Fab contains a complete antigen binding site, allowing it to still bind to its specific target. The third fragment is the Fc region, or fragment crystallizable. While the Fc fragment cannot bind antigens, it plays a crucial role in communicating with other components of the immune system, such as macrophages and natural killer cells. This division into Fab and Fc fragments is useful in research and therapeutic applications, helping scientists better understand how antibodies function in isolation. This image illustrates how antibodies recognize and bind to antigens on the surface of a pathogen, in this case, a bacterium. The red structures sticking out from the bacterial surface represent antigens, molecular markers that the immune system can detect. Each antibody binds specifically to a matching antigen through its variable region, shown here at the tips of the antibody arms. These interactions are highly specific, functioning like a lock and key. By coating the bacterium with antibodies, a process known as opsonization, the immune system marks the pathogen for destruction. This enhances the ability of phagocytic cells, like macrophages and neutrophils, to recognize, engulf, and eliminate the bacterium. This is a crucial part of the adaptive immune response, where antibodies not only neutralize pathogens, but also flag them for clearance by other immune cells. This slide highlights the 5 major classes of antibodies, each with a distinct heavy chain type and specialized function in the immune response. First is Ig, with a gamma heavy chain. Ig is the most abundant antibody in circulation and is critical for long-term immunity and pathogen neutralization. Next is IgM, featuring a mutation. It is the first antibody produced in response to infection and often forms pentamers to increase its binding strength. Ig, with a delta chain, is primarily found on the surface of immature B cells and plays a role in B cell activation. IgA, containing alpha chains, is found in mucosal areas such as the respiratory and digestive tracts. It plays a crucial role in neutralizing pathogens at mucosal surfaces and is commonly secreted in saliva, tears, and breast milk. Finally, Ig, with an epsilon chain, is involved in allergic responses and protection against parasitic infections. It binds to mast cells and basophils, triggering the release of histamine. Although all antibodies share a Y-shaped structure with variable regions for antigen recognition and constant regions that define their class and function, these differences in heavy chains give each isotype a unique role in immune defense. Immunoglobulin chains are folded into compact and stable protein domains. In this slide, we explore how immunoglobulin chains are folded into compact and stable protein domains. On the left, image shows a 3D structural model of an antibody. The antibody's heavy chains are shown in blue and yellow, while the light chains are in red and orange. These chains are folded into a specific arrangement of domains that contribute to the antibody's structural stability and function. On the right, image B provides a simplified schematic representation of the antibody structure. Each immunoglobulin molecule contains 2 heavy chains and 2 light chains. The variable regions, labeled as V sub H and V sub L, are involved in antigen binding and are located at the tips of the molecule. Constant domains, labeled C sub H and C sub, provide structural support. Disulfide bonds, shown here as connecting lines, help stabilize the folded structure of the chains. The hinge region allows for flexibility, enabling the 2 antigen binding sites to interact with antigens spaced at various distances apart. Together, this compact folding and modular design ensure both the structural integrity and functional adaptability of antibodies in the immune response. This slide shows how immunoglobulin chains fold into compact, stable structures that support antibody function. On the left, we see a 3D model of the antibody structure. Each colored segment represents a protein domain, folded into a stable, compact unit that contributes to the antibody's overall shape and strength. The diagram on the right breaks this structure down further. Each antibody arm includes variable regions, labeled V, which form the antigen-binding sites. These are critical for recognizing and binding specific targets. Adjacent to these are the constant domains, labeled NC for the heavy chains, and C less than sub greater than L less than slash sub greater than for the light chain. These regions provide structural support and mediate interactions with other components of the immune system. Disulfide bonds, shown as connectors between chains, stabilize the antibody's Y shape and keep the subunits aligned. Together, this domain-based folding system ensures that antibodies are strong, flexible, and functionally precise, allowing them to survive in diverse environments and effectively carry out their immune functions. An antigen binding site is formed from the hypervariable regions of a heavy chain V domain and a light chain V domain. In this figure, we explore how the antigen binding site of an antibody is constructed. The top graph highlights the variability across the amino acid residues of the light chain variable, or V region. Peaks in red indicate areas of high variability, known as hypervariable regions, or complementarity determining regions, CDRs. These include HV1, HV2, and HV3. Between the hypervariable regions are more conserved framework regions, labeled FR1 through FR4, shown in yellow. These framework regions serve as a structural scaffold that supports the hypervariable loops. In the middle panel, we see the three-dimensional structure of the light chain V region. The red loops represent the hypervariable regions, HV1, HV2, and HV3, also referred to as CDR1, CDR2, and CDR3. These loops project outward from the core beta sheet structure and are key to interacting with the antigen. Finally, in the bottom panel, we observe how the antigen-binding site is formed. It results from the combined contribution of hypervariable regions from both the light chain and the heavy chain variable domains. These regions come together to create a highly specific surface that recognizes and binds to a particular antigen. Antigen-binding sites vary in shape and physical properties. This figure demonstrates how antigen-binding sites can interact with antigens of varying shapes and epitope compositions. On the left side, we see a multivalent antigen containing different epitopes. Each distinct shape represents a unique epitope, and multiple antibodies bind to this antigen through their complementarity determining regions, with specificity for each epitope. On the right side, the illustration shows a multivalent antigen with a repeated epitope. In this case, the antigen has multiple copies of the same epitope, allowing identical antibody molecules to bind repetitively along the antigen surface. This structural diversity in antigen presentation allows the immune system to mount tailored responses. Whether recognizing a complex antigen with multiple unique sites or a repetitive antigenic pattern, antibodies adjust to match the antigen's spatial and chemical configuration. Understanding this variation is essential for designing effective vaccines and therapeutic antibodies that target specific antigen types with high precision. In this illustration, we examine the structural diversity of antigen-binding sites formed by the variable domains of the heavy chain, labeled VH, and the light chain, labeled VL. These domains come together to create a unique antigen-binding pocket. The first panel shows a simple round antigen fitting into a complementary binding groove, highlighting the principle of molecular complementarity, often described as a lock and key model. In the second and third panels, we see how antigen-binding sites can differ in contour, angle, and depth, accommodating a variety of antigen shapes, including flat or curved epitopes. Finally, the fourth panel shows a more complex interaction, where the antigen-binding site envelops the antigen in a snug, highly specific fit. This level of precision ensures strong binding through non-covalent forces such as hydrogen bonds, electrostatic interactions, and van der Waals forces. These structural variations are essential for the immune system's ability to recognize a vast and diverse range of antigens with high specificity. This figure compares two fundamental types of epitopes recognized by antibodies: linear and discontinuous. On the left, we see a linear epitope. The antibody binds to a continuous sequence of amino acids along the protein antigen's primary structure. These amino acids are arranged in a straight, unbroken chain, highlighted here in red. Linear epitopes are often preserved even when a protein is denatured, making them common targets in diagnostic tests. On the right, we see a discontinuous epitope, also known as a conformational epitope. In this case, the red binding sites are not adjacent in the protein sequence. Instead, they are brought into proximity by the protein's three-dimensional folding. This means the epitope only exists in the protein's native, folded conformation. These differences in epitope structure significantly affect how antibodies recognize and bind to antigens, influencing vaccine design, therapeutic antibody development, and diagnostic testing. Monoclonal antibodies are produced from a clone of antibody-producing cells. This figure illustrates the step-by-step process of generating monoclonal antibodies using hybridoma technology. The process begins with the isolation of B cells from a mouse that has been immunized with a specific antigen. These B cells are capable of producing antibodies against the antigen. Simultaneously, myeloma cells, which are immortal cancer cells, are also prepared. Next, the B cells and myeloma cells are fused together, forming hybrid cells called hybridomas. These hybrid cells combine the antibody-producing capability of B cells with the longevity of myeloma cells. The fused cells are then grown in a drug-containing medium. In this medium, only the hybridoma cells survive. Unfused B cells die naturally, and myeloma cells are eliminated by the drug. From the surviving hybridomas, scientists select the ones that produce antibodies specific to the antigen of interest. These are identified through screening assays. Finally, the selected antigen-specific hybridomas are cloned, producing a large population of identical cells that all secrete the same, specific monoclonal antibody. This powerful technology is the foundation of many therapeutic antibodies, diagnostic tools, and research reagents used in modern biomedical science. This figure demonstrates how flow cytometry is used to analyze and sort cells based on their surface markers, labeled with fluorescent antibodies. At the top left, a mixture of cells is labeled with different fluorescent tagged antibodies, each recognizing a specific cell surface protein. These labeled cells are then passed into a narrow stream through a nozzle where they move in single file. As the cells travel through the stream, they pass a laser beam. The fluorescent tags are excited by the laser, and the emitted light is detected and analyzed. The data collected allows for detailed analysis. In the one-dimensional histogram shown at the top right, cells are counted based on the level of a single marker, T-cell receptor. Peaks represent cell populations with different expression levels. The two-dimensional plot below that provides a more complex analysis, showing how cells distribute based on two markers, IgM and TCR. Distinct clusters represent subpopulations of cells. For example, 60% of cells are IgM high and TCR low, while 31% are TCR high and IgM low. Flow cytometry enables scientists to precisely sort and analyze millions of cells quickly, an essential step in isolating antigen-specific hybridomas for monoclonal antibody production. Monoclonal antibodies are used as treatments for a variety of diseases. This figure illustrates the four main types of therapeutic monoclonal antibodies, classified by the origin of their protein components: mouse, chimeric, humanized, and fully human. The mouse monoclonal antibody, shown in all blue, is entirely derived from a mouse immune system. While effective at targeting specific antigens, these antibodies are often recognized as foreign by the human immune system, leading to immune rejection. Chimeric antibodies, depicted in blue and yellow, are part mouse and part human. The variable regions that bind the antigen are mouse-derived, while the constant regions are human. This reduces the chance of rejection compared to fully mouse antibodies. Humanized antibodies are mostly human, with only the complementarity determining regions, those critical for antigen binding, retained from the mouse. This structure offers improved compatibility with the human immune system while preserving specificity. Finally, fully human antibodies are entirely generated from human gene sequences. These have the lowest immunogenicity and are best suited for long-term therapeutic use, especially in chronic diseases. Understanding these differences is essential in the development of biologic therapies, particularly in cancer treatment autoimmune diseases, and infectious diseases. Generation of immunoglobulin diversity in B cells before encounter with antigen. This figure shows the genomic layout of the immunoglobulin gene loci for both light and heavy chains, critical for antibody diversity. At the top, we see the lambda light chain locus, located on chromosome 22. It contains multiple V lambda segments in red, J segments in yellow, and C regions in blue. Each antibody light chain is generated by combining one V, one J, and one C segment. The kappa light chain locus is shown in the middle and is located on chromosome 2. Like the lambda chain, it consists of red V segments, yellow J segments, and a single blue constant region labeled C kappa. At the bottom is the heavy chain locus, located on chromosome 14. It is more complex, containing V segments in red, D segments in green, J segments in yellow, and multiple constant regions in blue. The heavy chain is formed by recombining one segment from each of the V, D, and J regions, followed by a C region. This genetic arrangement allows for immense combinatorial diversity, enabling the immune system to generate a vast repertoire of antibodies, each with a unique antigen-binding site. The light and heavy chains must both successfully undergo gene rearrangement to produce a functional immunoglobulin capable of antigen recognition. Random recombination of gene segments produces diversity in the antigen-binding sites of immunoglobulins. This figure explains how the diversity of antibodies is generated through random recombination of gene segments in both light and heavy chains. On the left, we see the process in the light chain. The germline DNA contains multiple V, J, and C gene segments. Through somatic recombination, one V and one J segment are randomly joined together. This forms the VJ rearranged DNA, which will be transcribed and translated into a functional light chain. On the right, the heavy chain undergoes a more complex process. The germline DNA contains V, D, J, and C segments. First, a random D segment joins with a J segment. This is known as DJ recombination. Next, a V segment is joined to the DJ pair, completing the VDJ recombination. Once these segments are recombined, the resulting DNA is transcribed, and the rearranged gene is expressed as a part of the complete heavy chain, including its constant region. This recombination process occurs in developing B cells and is a critical mechanism for generating the enormous diversity of antigen-binding sites necessary for the immune system to recognize virtually any pathogen. This table outlines the number of gene segments present in the 3 human immunoglobulin gene loci, which contribute to the structural diversity of antibody molecules. The first row shows the number of variable gene segments, which play a central role in determining the specificity of the antigen-binding site. The kappa light chain locus contains 34 to 38 variable gene segments, the lambda light chain locus contains 29 to 33, and the heavy chain locus contains 38 to 46. Only the heavy chain locus includes diversity gene segments, which provide an additional layer of variation. There are 23 diversity gene segments in the human heavy chain locus. These are not found in the light chain loci. The joining gene segments link the variable and diversity regions and contribute further to antigen-binding site diversity. The kappa light chain contains 5 joining segments. The lambda light chain contains 4 to 5, and the heavy chain contains 6. The constant gene segments determine the class or isotype of the immunoglobulin molecule. The kappa light chain locus includes 1 constant segment, the lambda light chain includes 4 to 5, and the heavy chain locus includes 9 constant segments corresponding to different immunoglobulin classes such as immunoglobulin M, immunoglobulin G, and immunoglobulin A. This organization of variable, diversity, joining, and constant join segments enables the immune system to generate a vast repertoire of antibodies, each with a unique antigen-binding site capable of recognizing diverse pathogens. This figure shows how recombination signal sequences flank the join segments in lambda light chain, kappa light chain, and heavy chain immunoglobulin genes. These signal sequences guide the somatic recombination process, where variable, diversity, and joining join segments are brought together to form functional antigen-binding regions. In both lambda and kappa light chains, a variable segment joins directly with a joining segment. Each segment is flanked by recombination signal sequences with specific spacer lengths that control recombination. In the heavy chain, the process includes an additional diversity gene segment. Recombination follows a strict order, diversity to joining, followed by variable to diversity, ensured by the 1223 base pair rule in the signal sequences. This precise recombination process allows for the generation of millions of unique antibodies, each capable of recognizing different antigens. This slide illustrates how random recombination of gene segments creates diversity in the antigen-binding sites of immunoglobulins. At the top left, we see a schematic of immunoglobulin join segments, V, D, and J. Each of these segments contributes to the formation of the variable region of the antibody. The recombination process begins with the enzyme complex RAG, which stands for recombination activating genes. These enzymes recognize specific recombination signal sequences flanking the V and J segments. In the second panel, RAG proteins bind to the recombination signals and bring the V and J segments together. Moving across the panels, we observe how RAG cleaves the DNA at the signal sequences, forming a hairpin structure. This brings the V and J regions into close proximity. Then, enzymes open the hairpins, trim the ends, and add nucleotides randomly, further increasing diversity. Finally, the segments are ligated together. This results in two distinct products: a signal joint, which is discarded, and a coding joint, which forms the functional VJ exon that encodes the variable region of the antibody. Through this process of VDJ recombination, the immune system can generate a vast array of unique antibodies capable of recognizing millions of different antigens. Recombination enzymes produce additional diversity in the antigen-binding site. This figure illustrates how recombination enzymes generate junctional diversity, a key source of variation in the antigen-binding region of antibodies. The process begins when recombination signal sequences flank join segments. A protein complex called recombination activating join cleaves these sequences, creating blunt DNA ends. Next, the DNA hairpins are opened asymmetrically, forming short single-stranded overhangs. These overhangs result in sequences called palindromic nucleotides. Terminal deoxynucleotidal transferase then adds random nucleotides to the ends of the join segments, further increasing diversity. The single strands pair imperfectly. Unmatched nucleotides are trimmed away by exonucleases. Finally, DNA synthesis fills in any gaps, and the segments are joined together to form a complete coding region. This random addition and deletion of nucleotides at the join junctions ensures that each B cell produces a uniquely configured antigen-binding site. Developing and naive B cells use alternative mRNA splicing to make both IgM and IgD. This figure illustrates how developing and naive B cells produce both immunoglobulin M and immunoglobulin D through a process called alternative messenger RNA splicing. Following somatic recombination, the variable, diversity, and joining join segments are brought together to form the variable region of the heavy chain. This is shown here as the VDJ segment, located immediately downstream of the leader sequence. The constant region gene segments, which follow the VDJ region, determine the class of the antibody. The first two constant region gene segments encode immunoglobulin M and immunoglobulin D, shown here in blue and purple. In naive B cells, both immunoglobulin M and immunoglobulin D are expressed on the cell surface. This is possible because the primary messenger RNA transcript includes both constant region sequences. Through alternative splicing, the cell can produce two different messenger RNA molecules, one encoding immunoglobulin M and the other encoding immunoglobulin D. This mechanism allows a single B cell to express two immunoglobulin classes with identical antigen specificity, supporting early immune recognition before class switching occurs. This figure compares how a single B cell can express either immunoglobulin M or immunoglobulin D by using alternative messenger RNA splicing. On the left, the expression of immunoglobulin M is shown. The Jun sequence includes the variable, diversity, and joining regions, followed by the constant region for immunoglobulin M. During transcription, all of these segments are included in the primary RNA. Through splicing, the messenger RNA retains the constant region for immunoglobulin M, resulting in production of that specific antibody. On the right, the same Jun sequence is used to produce immunoglobulin D. However, alternative splicing skips the constant region for immunoglobulin M and includes the downstream constant region for immunoglobulin D. This splice messenger RNA is translated into immunoglobulin D. In both cases, the variable region remains the same, meaning the antibody recognizes the same antigen. Only the constant region changes, allowing the B cell to produce two different immunoglobulin classes with identical specificity. Each B cell produces immunoglobulin of a single antigen specificity. Immunoglobulin is first made in a membrane-bound form that is present on the B cell surface. This figure shows the structure of the B cell receptor, which is the membrane-bound form of immunoglobulin found on the surface of immature and naive B cells. The receptor is composed of a membrane-anchored immunoglobulin molecule, shown here in blue. This membrane-bound form retains the same antigen specificity as its secreted counterpart, but includes a transmembrane region that allows it to remain anchored in the cell membrane. Because the immunoglobulin molecule itself does not signal into the cell, it is associated with two additional transmembrane proteins, immunoglobulin beta and immunoglobulin alpha, shown in orange and gray. These molecules transmit signals into the B cell when the receptor binds an antigen. Together, the membrane-bound immunoglobulin and its signaling partners form the complete B cell receptor complex, which is essential for antigen recognition and B cell activation. Secreted antibodies are produced by an alternative pattern of heavy-chain RNA processing. This figure shows how a developing B cell produces either a membrane-bound or secreted form of immunoglobulin M by using alternative processing of heavy-chain ribonucleic acid transcripts. On the left, the B cell generates membrane-bound immunoglobulin M. The rearranged gene is first transcribed into a long ribonucleic acid molecule. Through splicing, the messenger ribonucleic acid retains exons that encode a transmembrane region, resulting in a protein that remains anchored to the B cell surface. On the right, the cell produces secreted immunoglobulin M. In this case, splicing removes the exons encoding the transmembrane region, leading to a messenger ribonucleic acid that produces a soluble antibody. This version is secreted into the extracellular environment. This alternative processing allows B cells to switch from displaying surface receptors to secreting antibodies, which is essential for mounting an effective immune response. Rearranged V region sequences are further diversified by somatic hypermutation. This figure highlights how somatic hypermutation introduces additional diversity into the variable region of immunoglobulin genes after recombination has occurred. Following activation, B cells undergo rapid division in specialized regions called germinal centers. During this process, the enzyme activation-induced cytidine deaminase introduces point mutations into the rearranged variable gene segments. As shown here, the mutation frequency is highest within the region spanning the variable and joining gene segments. These mutations occur at a rate far higher than in typical genes and are tightly focused on the antigen-binding site. Somatic hypermutation allows B cells to produce antibodies with improved affinity for their specific antigen, a process known as affinity maturation. This is essential for optimizing the immune response over time. Through this mechanism, even after recombination, B cells can continue refining the precision of their antigen recognition. This figure compares the accumulation of mutations in the antigen-binding regions of antibodies 1 week and 2 weeks after primary immunization. On the left, we see the sequences from both heavy chain and light chain variable regions 1 week after immunization. At this early stage, there are relatively few mutations, and most antibodies are of the immunoglobulin M class. On the right, 2 weeks after immunization, a greater number of mutations are observed. These mutations are concentrated in the complementarity determining regions of both the heavy chain and light chain, especially in complementarity determining region 3, which plays a central role in antigen binding. By this time, some B cells have also undergone class switching and are producing immunoglobulin G. The presence of more mutations indicates that somatic hypermutation is actively refining antibody specificity through a process known as affinity maturation. This increase in mutation frequency over time enhances the immune system's ability to recognize and bind antigens with greater strength and precision. Isotype switching produces immunoglobulins with different C regions but identical antigen specificities. This figure demonstrates the structural transformation of immunoglobulin M from its monomeric to pentameric form. On the left, we see the monomeric form of immunoglobulin M, consisting of 2 heavy chains and 2 light chains. 5 of these monomers are linked together by a polypeptide called the joining chain, shown in yellow. On the right, the result is the pentameric form of immunoglobulin M, composed of 5 monomer units joined into a circular structure. This pentameric configuration greatly increases the molecule's ability to bind antigens and activate complement. Although the constant region changes during this structural shift, the variable region, the part responsible for recognizing antigens, remains the same. This preserves antigen specificity while enhancing the antibody's functional capabilities. The image on the far right shows an electron micrograph of pentameric immunoglobulin M, confirming its distinct 5-unit arrangement. This figure illustrates the molecular process of isotype switching, which allows a B cell to produce antibodies with the same antigen specificity but different constant regions. At the top, the rearranged variable, diversity, and joining gene segments are expressed with either the constant region for immunoglobulin M or immunoglobulin D. In response to signals, an enzyme called activation-induced cytidine deaminase targets specific switch regions located upstream of the constant region genes. Here, switch regions for immunoglobulin M and immunoglobulin gamma 1 are shown. This enzyme introduces breaks on both strands of DNA at these switch sites. A looping mechanism brings the targeted constant region in line with the variable gene segments. The intervening DNA is removed, and a new recombined locus is formed. As a result, the B cell now produces immunoglobulin gamma 1 with the same antigen-binding region, but a new constant region, giving the antibody different effector functions. Antibodies with different C regions have different effector functions. This figure compares the major immunoglobulin classes and subclasses, focusing on their structural and functional differences. Each antibody class is defined by its unique heavy chain type. For example, immunoglobulin M has a mu heavy chain, while immunoglobulin G subclasses have gamma 1 through gamma 4 chains. Immunoglobulin A has alpha 1 and alpha 2, and immunoglobulin E has an epsilon chain. The molecular mass varies significantly across classes. Immunoglobulin M is the largest at approximately 970 kilodaltons, while others range from 146 to 188 kilodaltons. Their serum concentrations also differ. Immunoglobulin G1 is the most abundant, followed by immunoglobulin A. In contrast, immunoglobulin D and immunoglobulin E appear in much smaller amounts. Finally, half-life in serum shows how long each antibody remains active in the bloodstream. Immunoglobulin G1 has the longest half-life at 21 days, while immunoglobulin E persists for only 2 days. These differences in structure, stability, and abundance reflect the distinct effector roles of each antibody type in the immune system's response to pathogens. This figure summarizes how each immunoglobulin class contributes to different immune functions based on its constant region. The chart compares several key activities, including neutralization, opsonization, natural killer cell sensitization, mast cell and basophil sensitization, complement activation, transport, and tissue diffusion. Immunoglobulin G1 shows strong performance in neutralization, opsonization, and placental transport. Immunoglobulin A, particularly in its dimeric form, excels in epithelial transport and mucosal immunity. Immunoglobulin E is uniquely specialized for sensitizing mast cells and basophils, playing a central role in allergic responses and defense against parasites. Immunoglobulin M is a powerful activator of the complement system, while immunoglobulin G3 combines complement activation with strong opsonization and neutralization. Some functions, such as diffusion into tissues or placental transfer, are limited to specific immunoglobulin subclasses, reflecting differences in size, structure, and receptor interactions. Together, these differences allow the immune system to deploy the most effective antibody type for each stage and site of infection. This figure illustrates the structure and function of dimeric immunoglobulin A, the primary antibody found in mucosal secretions such as saliva, tears, and intestinal fluids. At the top, we see two monomer units of immunoglobulin A joined by a joining chain, shown in yellow. This polypeptide enables the formation of the dimer and facilitates transport across epithelial cells. Dimeric immunoglobulin A plays a vital role in mucosal immunity by neutralizing pathogens at points of entry before they can invade tissues. The electron micrograph at the bottom confirms the dimeric structure, showing two antibody units linked together. This configuration enhances the antibody's stability and effectiveness in mucosal environments, where immune protection must be both strong and long-lasting. This figure demonstrates the remarkable flexibility of the immunoglobulin G molecule, which is essential for its dual function, binding to both antigens and immune system receptors. The immunoglobulin G structure includes two antigen-binding arms, known as the fragment antigen-binding regions, and a constant region known as the fragment crystallizable domain. The hinge region allows for multiple types of movement. The arms can wave and rotate, the tail can wag, and the elbows can bend, adjusting the position of each arm. This structural flexibility enables immunoglobulin G to bind antigens that are spaced variably on the surface of a pathogen while also engaging with receptor proteins on immune cells. Such dynamic movement enhances immune complex formation and promotes effective clearance of pathogens. The four subclasses of IG have different and complementary functions, antibody structure, and the generation of B-cell diversity. This figure illustrates the structural differences among the four subclasses of immunoglobulin G, focusing on their hinge regions. Each antibody consists of two light chains and two heavy chains. The antigen-binding sites are formed by the variable regions at the tips of the light and heavy chains. What differentiates immunoglobulin G1, 2, 3, and 4 is the structure and flexibility of the hinge region, which connects the arms to the body of the molecule. Immunoglobulin G1 has a moderately flexible hinge, allowing for efficient antigen binding. Immunoglobulin G2 has a shorter, more rigid hinge, which limits flexibility but enhances resistance to proteolysis. Immunoglobulin G3 has the longest hinge, granting high flexibility but making it more susceptible to cleavage. Immunoglobulin G4 has a unique hinge that allows for exchange of half molecules, a feature that contributes to its anti-inflammatory role. These structural variations allow each subclass of immunoglobulin G to perform distinct immune functions while sharing the same overall architecture. This figure compares the four subclasses of immunoglobulin G across multiple functional and structural characteristics. Immunoglobulin G1 is the most abundant subclass, making up between 45 and 75% of total immunoglobulin G in serum. In contrast, immunoglobulin G4 is the least abundant. Hinge length varies significantly. Immunoglobulin G3 has the longest hinge, consisting of 62 amino acids and 11 disulfide bonds, contributing to its high flexibility and sensitivity to enzymatic cleavage. Susceptibility to proteolytic cleavage increases with hinge length. Immunoglobulin G3 is the most vulnerable, while immunoglobulin G2 and 4 are more resistant. The half-life of immunoglobulin G subclasses also varies. Immunoglobulin G1, 2, and 4 all persist for about 21 days, while immunoglobulin G3 has a shorter half-life of 7 days. In terms of immune function, immunoglobulin G3 is the strongest activator of the complement system. Immunoglobulin G1 also contributes significantly, while immunoglobulin G4 has limited ability. Each subclass also shows distinct patterns of antigen response. Immunoglobulin G2 is more responsive to carbohydrate antigens, whereas immunoglobulin G4 is more responsive to allergens. This figure shows a unique property of immunoglobulin G subclass 4, its ability to become functionally monovalent in the circulation. On the left, we see two typical immunoglobulin G4 molecules. Each has two identical antigen-binding arms. In the bloodstream, a process called Fab-arm exchange occurs. This allows the antibody's half molecules to separate and recombine with halves from another immunoglobulin G4 molecule. As shown on the right, the result is a single antibody with two different antigen-binding arms. Since each arm binds a different antigen, the molecule is unable to cross-link antigens effectively. This property makes immunoglobulin G4 functionally monovalent and significantly less pro-inflammatory than other immunoglobulin G subclasses. This feature contributes to immunoglobulin G4's unique role in dampening immune responses and promoting immune tolerance. This figure summarizes the sequential steps in the generation of antibody molecules, showing both the light chain and the heavy chain pathways. On the left, we see the process for the light chain. It begins with germline deoxyribonucleic acid, where multiple variable, joining, and constant joined segments are present. Through somatic recombination, one variable and one joining segment are selected and joined, forming the rearranged deoxyribonucleic acid. This rearranged join is transcribed into primary ribonucleic acid, which still contains introns. After splicing, a mature messenger ribonucleic acid is produced. Translation then generates a functional polypeptide chain, which folds into the final immunoglobulin light chain structure. On the right is the process for the heavy chain, which includes an additional step: the joining of diversity gene segments between the variable and joining regions. The heavy chain gene undergoes variable, diversity, and joining recombination, followed by transcription, splicing, and translation, ultimately producing the complete heavy chain, which includes constant domains responsible for class and function. The final antibody molecule is formed by pairing one light chain and one heavy chain, with the variable regions determining antigen specificity and the constant regions defining effector function. This table summarizes the major genetic changes that occur in B cells during their development and activation, and whether those changes are reversible or irreversible. First, variable region assembly occurs through somatic recombination of joined segments. This process is irreversible and sets the foundation for antigen specificity. Second, junctional diversity is generated by the imprecise joining of gene segments, introducing additional nucleotides. This further diversifies the antigen-binding site and is also irreversible. Third, the assembly of transcriptional control elements brings promoter and enhancer regions closer together to enable proper join expression. This change is permanent. Fourth, co-expression of immunoglobulin M and immunoglobulin D on the B cell surface is achieved through alternative ribonucleic acid splicing. This step is reversible and regulated. Fifth, when a B cell is activated, it shifts from producing membrane-bound antibodies to secreted antibodies using additional splicing patterns. This too is reversible and regulated. Sixth, somatic hypermutation introduces point mutations in the variable region genes to improve antibody affinity. This is irreversible. Seventh, isotype switching changes the constant region through recombination, allowing the B cell to produce different classes of antibodies. This process is also irreversible. Together, these genetic mechanisms provide the flexibility and precision needed for an effective, adaptive immune response.